how long it's going to take, so we shall see. It's got some very, very awkward, complicated bits in it, especially if, like me, you don't know any biochemistry. But we'll get over the hard bits and into the easy bits. What I want to talk about essentially is the role of the liver and other things in dealing with blood glucose concentration. Now, blood glucose concentration is incredibly important. If it goes wrong, life gets very complicated. So we'll talk about the inputs and outputs to blood glucose, and that's the really complicated bit which you just have to sweat through because you need to understand that bit. Again, if you're doing lots of biochemistry, you probably understand it already. We recognize two states, the fed state and the fasting state. If you're fed, as I hope you are now, then you will have a rising blood sugar concentration because you're absorbing sugar from the gut and other things as well, but essentially blood glucose is tending to rise. That has to be kept within narrow limits. So that's the characteristic of the fed state. The fasting state, you are not absorbing, it's post-absorptive, you are not absorbing blood sugar into the blood, or indeed anything else, and so you're using the stores. So you're using stored glucose, stored fats, stored amino acids. So that's the difference between two states. We'll see about those. Then we'll go on and talk about the regulation of these processes, essentially by insulin and glucagon. We may mention a couple of other hormones that do something about this. We have mentioned some when in the hormone lecture way back in the dim and distant, which see. And then lastly, I spend most of the sort of last third of the lecture, I hope, possibly tomorrow, talking about diabetes, by which I mean specifically diabetes mellitus. That's to say, sugar diabetes, as it's called in lay parlance. The different types, the different effects, both in the short term and the long term. And lastly, because I was interested in it, I thought I would talk a little bit about diabetic coma, since it seems paradoxical to me. So I thought if I can explain it to myself, I can probably explain it to you. So we'll all finish up in a diabetic coma. It'll be great fun. But the least amusing part of diabetes is diabetic coma. This diagram from Sherwood, I'm going to repeat this diagram, and I'm going to present it, the same information in words in the next slide, which is very, very ugly and very dense, and it's a Tuffrey original. But essentially, we can think of blood glucose as a compartment. You know how we physiologists like compartments? It has inputs and it has outputs. That's the key. Now, you've got to work out what the inputs are and what the outputs are. Then in all the situations that we're going to describe, you want to work out which way do I want things to go. Do I want blood sugar to be added to or subtracted from? And then once you've learned the inputs and outputs, you can make all the equations go the right way. So that's the core of the lecture. Input, output, and a bit of teleology, and you're away. So that's how it works. So let's look. Here's blood glucose concentration. Oops. Here's blood glucose concentration in the middle, and you've got inputs to blood glucose concentration on the left of the figure. So glucose absorption from the digestive tract. That's easy, and that's the variable. That's fasting and fed. Glucose production from the liver, and there are essentially two processes. Breaking down of glycogen, right? Glycogenolysis, breaking down of glycogen. Glycogen is stored glucose. It's the storage molecule for glucose. So that's if you visualize that as a reversible equation, then that makes it all work. So you can break down glycogen to liberate glucose into the bloodstream, or there is another process here called gluconeogenesis, forming new glucose. And that's formed from amino acids. Those of you who know and care about biochemists could probably give us a 40-minute disquisition on that. I'm not going to. It's a black box. That's all we need to know, certainly for the rest of us. It's a bot. You can produce glucose from amino acids. That's biochemistry, folks. Clever. But that's new glucose. It's not from stored glycogen. So that's a kind of emergency top-up. On the output side, glucose goes into cells. Essentially, that's what insulin does. It increases the absorption of glucose into the cells of the body. A very big compartment. A third of total body water is in the cell. So it's a big compartment. And it's used there for utilization, for energy production, biochemical work and so on, and of course for storage, usually in the form of glycogen, particularly in the liver cells and skeletal muscle. They're the main storage areas. And also as fats, and that's in fat cells, mostly. You can also get rid of blood glucose by excreting it by the kidneys, but that's pathological. So you only find sugar in the urine if you're just unable to cope. So the kidneys can't cope. That's pathological. So the little dipstick to see how you're excreting sugar is a pathological test. So it only happens when you completely overwhelm the absorptive capacity of the kidney. So that's essentially it. That's the inputs and outputs. We'll have to look at a bit more detail. Now, this is the key thing what you have to remember. You have to keep a reasonable supply of blood glucose at all times, because it's the sole fuel, virtually so, 99% so, for the central nervous system. So if blood glucose falls, central nervous system function diminishes. This causes all sorts of effects. If you've got any friendly diabetics who get mild hypos, or indeed serious hypos, the effects are a bit like them getting drunk very quickly. They become disinhibited. That's to say, they get rowdy and argumentative and stroppy. And that's the time to get in the old glucose, a drop of Lucozade and a chocolate bar or something quick, because they're going into a hypo. So recognised state. They get very cranky, or may do at that stage. And dangerous to be in charge of machinery and driving and all those things. So sole fuel for central nervous system. That's crucial. So you've got to keep blood glucose levels up. What happens when it gets too high, we'll come back to later on, but it's a very serious issue if it gets too high. So inputs and outputs, feeding and fasting. That should be fed and fasting states. Little finger dithered.
Can you turn down the light a bit?
Oh, sorry, yes, certainly now, yeah. Better? Thank you. So our aim in all of this is thinking teleologically, is to maintain blood glucose concentration. Okay, so that's the aim, within narrow limits. And as you'll see, we've got something that depresses it, namely insulin, and we've got something that stimulates it, namely glucagon. So it's what I used to call a push-pull system before I was told not to. So you're squeezing blood glucose levels into a narrow band. Now this is the horrible slide. And we're going to look at the biochemical process, what happens in the liver and what happens in the blood. Now remember here that the core value that we're looking at is what happens in the blood. So you've got glycogenesis, production of glucose. Sorry, production of glycogen, obviously. Glycogenesis. Glucose is stored in the liver as glycogen. In other words, it is being removed from the blood. It's being taken from the blood, put into the liver, stuck together to make a storage molecule, and that reduces good shot. That wasn't very far though. Glycogenolysis is the reverse process. So they go in pairs. This is the easy bit. So you don't even have to remember their names if you remember the processes. Glycogenolysis, the lysis meaning breaking down. Glycogen to glucose, so blood glucose goes up. So just think in terms of storage molecules, glucose concentration. Gluconeogenesis doesn't go in pairs, it's a one-off. It's the forming of new glucose, neo meaning new. Important source of new glucose. Amino acids go to glucose, so blood glucose goes up, occurring in the liver. So we'll see how that's switched on later. We'll have a look at a couple of marathon runners and see how they work with these processes. Other reactions, what else have you got? You've got fats and you've got lipids, so you can work those out. Protein synthesis, you've got protein synthesis in the liver, takes out the amino acids from the blood. So it's the same pool. Opposite process, protein breakdown, proteins go to amino acids, blood amino acids go up. So they're available for metabolism. Fat synthesis in the liver, fatty acids to triglycerides, blood fatty acids fall, and the reverse process. So that's the summary of all that biochemistry. Some of you will know a lot more than that. So now all you need to think of then is you've got those, 1, 2, 3, 4, 7 processes. 7 processes, which you ought to be able to make up anyway, because you've got glycogen, fats, proteins. So input and output. So that doubles it, plus gluconeogenesis. So that's the model. Then all you've got to do is work out which way they've got to go in order to sustain blood glucose concentration. So when you're fed, seems a long time ago since my breakfast, but anyway, when you're this is the same figure as before. It seemed not to have carried its legend over. When you're fed, you've got lots of glucose sloshing around because you're absorbing glucose essentially from carbohydrate. It's your major energy source. That's what you're using to keep you going, to keep your metabolism going, to do anything else you're doing. Glucose, there's plenty of it. At the same time, because you've got plenty of it, you're storing some of it for later in the form of glycogen in the liver and skeletal muscle. If you've got excess, then you're laying down extra triglycerides in your fat deposits. So that's that. So you can see how each process is affected here. So all the equations is this process is an input. This process is going to be an output. You're switching off gluconeogenesis. You don't need any more. So that's switched off. That's inhibited. And this process is not glycogenolysis, but stored glycogen. So that's an output. So there we are. We can see it's a special form of that one. Urinary excretion won't come in because that's pathological for most of us. Compare that with the fasting. Now you've got no free supply of glucose from the gut, so you've got to maintain blood glucose concentration to keep your brain happy. Central nervous system, all of it. So you're forming glucose, and you form it by breaking down glycogen, as in that one, glycogenolysis. Here we are, glycogenolysis, blood glucose. And you've got gluconeogenesis, forms glucose from amino acids in there. So that's all from left to right. That's all forming glucose, because we haven't got any more. If you run out, then you've got a phenomenon known as glucose sparing. Very clever little trick, this. This means many of the tissues switch their metabolism away from glucose and use fats and amino acids as the primary fuel supply. That's very clever, because you're maintaining the glucose for the one area of the body which can't use anything else, namely the brain. So glucose sparing is important. In fact, glial cells in the brain and the nervous system can switch their metabolism very neatly, so it's always opposite, if you like, or complementary to the neuron metabolism. So if the neurons are using glucose, they switch away from glucose to spare it all the time if there's a lack. Okay, so those are the core mechanisms. So you can work through those. Now, these next two slides are simply a boring restatement of what was in the big slide. So in the fed state, you've got a surplus of amino acids. You're breaking down proteins in the gut. You're absorbing amino acids of various kinds, and those are being passed to the various cells in the body, and they're being synthesized into proteins, mostly for active proteins, possibly for the storage. In the liver, you may have amino acids for energy, that's keto acids, not normally at that stage because you've got glucose around, and you may have triglycerides going to adipose tissue. We'll come back to keto acids again. Again, the biochemists, those of you who do lots of biochemistry, may know a lot more about keto acids, but they'll come back to haunt us when we talk about diabetes. In the fasting state, you've got muscle. It generates, it's a storage of glycogen, so it will generate that, and it will break down proteins if you're really fasting. In starvation, that's what you do. Liver will form glucose by gluconeogenesis. So you're pushing down, you've used up the blood glucose, you've got to replenish it, and you switch on gluconeogenesis. And again, we'll see the effects of that on extreme stress in a couple of marathon runners, or at least in one of them, where one of them doesn't cope very well. Lipids doing exactly the same. Fat cells, when you're fed, you're forming triglycerides and storing them. And in the liver, triglyceride synthesis, and eventually that goes on to the adipose tissue for storage. We've seen a little bit elsewhere about that. Fasting cells, it's the opposite way around. The fat cells liberate their fat, breaks down, goes into the circulation. It's used via the blood to most cells and is used as an energy supply. So fat is the reserve for energy. So if you eat nothing, I do not recommend that you eat nothing, but if you eat nothing, you will eventually break down your fats, and you will eventually, if you're not careful, break down your proteins as well. That's not a good thing to do. So free fatty acids go to ketones, and again, we'll come back to those. Now, the regulation, the role of insulin. Now, insulin is dead easy, provided you remember which way around it works. It's secreted after a meal in order to keep blood glucose down within normal limits. So it's squashing blood glucose down. That's what it does. It acts to prevent the rise. So as soon as glucose starts to rise, it's detected and it acts to prevent any further rise. It has multiple effects. But that's the main effect there, is to increase glucose uptake by all of the cells of the body. So it acts on all cells. Remember, it's a hormone secreted into the blood, so it reaches virtually all the cells in the body very quickly. And it switches them up and gets them to take up blood glucose very rapidly. Now, let's have a look at that. Here's our little classic feedback loop. Blood glucose concentration rises. Major control mechanism, B cells. You do not need to remember that they're beta cells. Secrete insulin, and it does all of those things. It switches all of those processes in the right direction to reduce blood glucose. Notice that little bit of teleology there, the right direction. So blood glucose falls, blood fatty acids fall because protein breakdown is inhibited, protein synthesis is encouraged, blood amino acids, fats synthesis and breakdown inhibited. You have to do both, encourage synthesis and inhibit breakdown, right? Two processes each time. And the same with those, fuel storage. So those are complementary. So you get a blood sugar concentration, and there we are, that's false. Stress will do the same, so it's a fight and flight reaction. That inhibits. Parasympathetic stimulation, so when all is happy, your blood glucose level can be allowed to drop a bit. So there we are, that's all of those processes. There are lots of other insulin responses other than the glucose uptake, and you've seen the fatty acids and the proteins, they're all there as well, they act. Right, now, so these are the other uptake. This is a little bit more detail on how the actions go. So the key aim is to get glucose out of the blood. Same story over and over again. Increases cellular uptake by increasing the activity and numbers of that co-transporter. GLUT5 is there. Increases the hepatic phosphorylation of glucose. Now, you wouldn't invent this one if you were just doing this as a black box. You might if you were very clever. It's a bit like our triglyceride synthesis during digestion inside the gut cells. It's a way of maintaining the gradient of glucose into the hepatocytes, the liver cells. So what happens here is the glucose gets inside the liver cells and it sticks a phosphor compound on the side or group on the side, and that keeps the concentration for glucose high, or the gradient high, so it still encourages it to come in. So you promote glycogenesis, that's to say the formation of glycogen, so that's also sucking in glucose into the liver cells all the time. On the supply side, decrease the supply by inhibiting gluconeogenesis and obviously glucogenolysis. Glycogenolysis is inhibited. Can't go both ways at the same time. So affect the supply side and the output side. And much the same happens for fat and protein, so squash those two together. So it's acting also to reduce the fat and the proteins in the blood because they're all potentially fuels. Fat out, glucose entry to cells, formation of fatty acids, promotes that fatty acid from glucose. Is that what we want to do? Yes, we do. Yes. Promotes the fatty acids into cells. So again, drawing it out of the blood into the cells. Reduce the fat formation, so it inhibits the breakdown of adipose tissue to liberate fats, as you would expect, I hope. Same principles with protein. I don't think I'll bother saying all of that. Active amino acid entry into the cells. Protein synthesis, then drawing amino acids out of the blood. Inhibit the other side of the process. Good. Right, now, let's get a two-hormone system. We've had one of these recently, I think. Insulin and glucagon oppose one another. And here's the diagram. So, blood glucose rises, and as we've seen before, the beta cells produce insulin, it rises, and that tends to depress blood glucose levels to normal by increasing essentially cellular uptake and all the other little bits as well. Glycogen, on the other hand, is produced from the alpha cells, which you needn't remember, and it goes down, it falls. Glucose rises, glucagon falls. So that it switches off the supply side. So glucagon is the one that comes in when glucose levels fall, glucagon switches in. So you reverse the process. If you reverse that arrow, you reverse the signs here and the signs here, and you get to restore it. So the combined effect is to squash blood glucose concentration within that nice narrow range. Crucial. Right, we've looked at everything inside. Let's look at the outside for a few moments. What affects food intake? Apart from being just greedy, as some of us are, there are different kinds of regulators. There are short terms and the long terms. And the short terms are much as you might expect them to be. If you've got a full gut, then you don't eat any more. The signals go back up to the hypothalamus to indicate satiety or fullness, and your appetite is depressed. Now, having very large brains, we can override that, and we sometimes do. So we eat far too much, and we're very good at eating far too much, almost a design fault, you might say. So we can override it because we have so much choice and so many other things going on. So we can override our satiety centre and fill ourselves up. If you're using glucose, then you may want to top. You may get a feeling glucose is too low. You may want to top up. Insulin affects appetite, and also do ATP turnover, and CCK has an effect. So all of these active hormones, which are being produced during the fed state, are depressing appetite, as you might expect. In the long term, you actually have a signal from your fat stores. So your fat stores release a little hormone called leptin, and that gives a measure of the total fat mass in your body. And that acts to the central nervous system, primarily through the hypothalamus, and that switches off feeding. So if you had anti-leptin or a leptin deficiency, what would happen? You wouldn't have an indicator to tell you what your fat mass is, and your feeding pattern wouldn't be inhibited. And there are, would you believe it, there are genetic rats and mice which don't have leptin, can't produce leptin, and they're called obese mice. And they're wonderful. Their little legs stick out the side and they can't walk very far. They just sort of sit there with their little legs out like that. But you can also get rats to eat far too much just by giving them lots of things to eat. Lots of different things. Instead of the ordinary horrible food that rats get in labs, you give them chopped up bits of Mars bar and all sorts of other exciting things. They'll keep on eating till further orders and generate obesity that way. Sound familiar? Yeah, it does, doesn't it? Right. Let's have a talk about diabetes for a bit. It came as news to me that there are different units for measuring these things, but I suppose I should have known this. So there are different kinds of units around, and you need, I suppose, to be familiar with them. Normal blood glucose concentration. Notice we've been talking about blood concentration and not plasma concentration all the time. So it is blood concentration that's measured. We don't spin down. That's the normal range. Around 90 milligrams per deciliter. For some reason, medics like deciliters, unlike normal human beings who like litres. But anyway, it gives you the right kind of number. 2 decimal places, 2 figures before the decimal place, usually. So around 90 is the normal value for that, with a range around it. So you are hyperglycemic, that is to say you have clinically, statistically, both of those, you have a pathological condition if you have more than 120 milligrams per deciliter. So that's pathological. This is dangerous. This is going to cause problems. You are hypoglycemic if you've got below 60 milligrams per deciliter. You are now confused, right? And your poor little central nervous system is going to struggle. Right, now, black box thinking. Insulin is the controlling mechanism here, or the primary controlling mechanism. You can have two types. We call them type 1 and type 2. You can have, as in the case of type 1, you can have a problem with producing insulin. If you can't produce enough insulin, that's type 1 diabetes. Insulin dependent diabetes mellitus, type 1. Right? Insulin dependent diabetes mellitus. Now, I will never give you the one name without the other name to try and give you a bit of a clue. But both terms are used because type 1 doesn't really tell you anything. It's insulin dependent. The beta cells are not producing insulin or not sufficient insulin. And usually that's an autoimmune condition. In other words, the immune system of the body, for some reason best known to its little self, has decided to attack the beta cells and you can't produce enough insulin. These things happen. That's the price you pay for having an immune system, I suppose. Characteristically, these occur in young persons, juvenile onset, a bit like pimples, and only about 10 or 15% of total cases of diabetes. It's a relatively rare condition, fortunately. So the treatment is insulin replacement, essentially. So usually insulin injection. Technology's got pretty good there. Modern techniques, islet cell implantation, and latterly experiments with stem cell implantation. You can collect stem cells from an individual and you can persuade them that they ought to be beta cells and you can get them into the right place. Fine, they'll all work very nicely. That's still experimental, but that's probably where the future lies. And of course, type 1 diabetics need to monitor their blood glucose concentration very carefully, and there's quite sophisticated apparatus for doing so. And if you have any friendly diabetics in your place, you will see them do that. They monitor their blood glucose concentration and they give themselves appropriately graded doses of insulin. It's all really rather sophisticated nowadays. Type 2, there are experiments with pumps and all sorts of feedback systems and all of that, but that's very much experimental. Type 2, non-insulin dependent diabetes mellitus. That's what NIDM means. Non-insulin dependent, and that's due to not a lack of insulin, but a failure of the target cells in the body to respond to insulin. So there's an impaired response. It's the only mechanism you can have when you think about it, either not enough stimulus or an inability to respond to it. Characteristically adult onset, typically pretty late in life, with one kind of exception. And that's the vast majority of cases. So there's going to be a lot of business here for medics here. You're going to be managing people, particularly those in the West, because 80% of such people are obese. Obesity is highly linked with diabetes. So probably due to a kind of desensitizing, because overeating, inability to metabolize normally will result in excess insulin production or a lot of insulin production, and it appears that the cells get desensitized to insulin, and that then sets up a feedback and you get obesity. So you've got to manage that by weight control. And in fact, I had an article where there's a serious discussion of stomach stapling as a treatment for diabetes. So in other words, you reduce intake. It's a pretty drastic thing to be doing. The proponents think it works. Exercise is very effective because exercise will increase the sensitivity to insulin. Now, if you're obese, exercise is very difficult, so both socially and practically. So it's kind of difficult to start, but there are increasing tendencies to get people to exercise much more because it does increase sensitivity. It may assist with weight loss. So if you can staple their stomachs and get them moving, you've got a fair chance you might do things. there's an odd little effect here. I am certainly not, repeat, not going to ask you an exam question of any form on this particular slide, because I think it's quite a difficult concept. So if you don't get it, let it go, is my advice. But it's an interesting little idea. Remember that insulin increases the permeability of cells to glucose. Right. That's what happens. So, paradoxically, it has an odd effect here that insulin, well, it's not a paradoxical effect at that point. If you haven't got enough insulin, then you don't get permeability of the pancreatic alpha cells to glucose. So they're the cells that are going to produce glucagon. So they register the glucose level as being low because it's not getting into the cells. They're recording it as being low. Although it's not low, it's high. So it perceives, I may use that term, a fall in glucose concentration, and you get glucagon production. And that tends to push up the blood sugar level even first. And I put in a couple of jawbreakers there because sometimes I like them. It exacerbates the hyperglycemia. It makes it worse. The tendency is the same. So that's a paradoxical rise in glucagon in that condition. So it's a very odd one. Again, if you don't worry, if you don't get it all, don't get all the steps there, don't worry about it. I'm certainly not going to ask you to. Although, it's a sort of question the anatomists like, isn't it? You ask the last 1% of knowledge on the principle that people must know the other 99%. There's no evidence for this. Right. There's a nice old-fashioned diabetic giving herself a little jab in the arm. Nowadays it's done in a much more sophisticated manner, that, and much more discreetly. If she shoots up like that nowadays, she'll be arrested probably. So there we are. She's not looking for a vein. She's all right. It's IM. There you are. Right. Now, let's have a look at the effects. There are short-term effects and there are long-term effects of type 1 diabetes. So this is a quick summary, and then we'll go on and look at the other aspects. So lipolysis is increased. That's the breakdown of fats in the stores. Fatty acids up. Ketosis. That's produced by using fats for metabolism. They are keto acids. So if you've got lots of acids being produced, that's going to lead to acidosis. Now, when I say acidosis, that's like labeling a drawer in a filing cabinet for you. You actually know lots about acidosis. You've done it in respiration, you've probably done it in relation to renal function, or you will, and biochemistry may have talked about lots of that too. So behind that is the whole acidosis story. So that leads to metabolic acidosis, and that can lead to all sorts of nasty conditions, which we'll look at in a bit. You inhibit protein synthesis, so that means you get impaired growth and repair. Typically, type 1 diabetics tend to be very scrawny. They can't maintain protein mass. As well as not being able to maintain glucose, they can't maintain protein mass, and that leads to general weakness and being fairly thin. Low body fat as well. Oh, didn't know it was going to do that. Don't worry about that. That's a very complicated picture, which says the pathway is down here through this mechanism, glycation or glycation, comes down there, and the end result is damage to the small blood vessels. Now again, biochemists may find that terribly fascinating. You get all sorts of things. You get hyperglycemia, there are lots of effect, free oxygen radicals in there, and there's a whole set of pathways, and that leads down to effects on the blood vessels, both at the micro level and the macro level. The macro level we've already seen something about, the production of thrombi. So that's part of that. And that can be due to changes in collagen, which encourages storage of all sorts of nasty things like low density lipids and cholesterol and so on. So the possible effects there are glucosuria. So high rising glucose may appear, you may get sugar in the urine, you will get an increased volume of urine. Why? You've got excess of glucose, yeah, in the blood, in the plasma filtrate. So that tends to hold water in the filtrate. That means urine volume will increase. Even though you actually don't need it to increase, it will increase because of that osmotic effect of the excess glucose. That will lead to dehydration. Dehydration, among other things, leads to reduced blood volume, and you know what blood volume does to blood pressure. If blood volume falls, blood pressure will down, and you get collapse and death. Very spectacular. I mean, not the death bit, I mean the collapse bit. I've actually seen it in a diabetic in a crisis where our friendly neighbourhood nurse came in, took a blood pressure of a supine subject who was quite in a bad way, and said, stand up. Well, didn't get fully erect before falling down a little heap on the bed. This time, that's when you dial, whichever number it is, you now dial for an ambulance. This is crisis time. But you do get damage to blood vessels, and this explains nearly all the serious consequences of diabetes. Damage to small blood vessels. We've seen atherosclerosis, laying down of collagen, cholesterol plaque and so on, leading to thrombosis and emboli. You get reduced flow in various areas that will cause tissue damage. Stroke, heart disease, and renal failure. Biggest single cause of renal failure leading to a requirement for transplant. Biggest single cause of blindness is diabetes in these islands, hereabouts. And overall impaired repair mechanisms. Because of the reduced circulation, especially in the periphery, you get impaired repair mechanisms, you get tissue damage, and quite minor lesions which wouldn't bother the likes of us, like ingrowing toenails, small cuts on the feet and hands, particularly the feet, heal very, very slowly, particularly in type 2 diabetes, where you may have elderly patients. Their circulation is poor, they're not very active, they do too much sitting around, probably malnourished anyway, and they get these poor repair mechanisms, and they get gangrene. Gangrene, associated with Scott's expedition to the Antarctic and things like that, and serious mountaineers. Not with little old ladies sitting in little council flats around the place, but that's where gangrene occurs, leading to amputation and serious consequences. So, nasty stuff. This pair of pictures looks really quite horrible, and the reason it looks quite horrible is because I whisked it into whatever it is you whisk it in, Photoshop, and slapped all the little levers in Photoshop out to the right hand side, because the original of this picture from Stanfield and Gurman and Stanfield, is actually done in such pale colours that you can't project it, which wasn't very clever of them. So I've zapped everything up there so it looks a bit horrible. The purpose of this pair of figures is to illustrate the changes in the mechanisms depending upon fuel resources in particular conditions. We've got here two marathon runners. What do you know? They're called Jane and John. Which is the stupid one? You got it. John. So here we are. This is the male runner. This is his glycogen reserve here, and that's his blood glucose. As he runs along through the race, you can see that his blood glucose is providing quite a lot of the fuel, but glycogen is producing 50% of the energy. This is his actual glycogen usage as he goes in here, and you can see he hits the wall at about 20 miles. He's gone too fast. He's run out of fuel. Look and see what else happens though. Fatty acid increases, and as he gets towards the end, gluconeogenesis is cut in. Glucose has fallen to a relatively low level, and he is now using amino acids to bump up his glucose level. So that's the contribution, a greater contribution to his total energy output. Whereas dear Jane, being sensible, has run at a better pace, and she hasn't run out of glycogen. She hasn't hit the wall. Of course, the silly woman could have gone faster and got a better time, because she should finish ideally with her glycogen down there and fall out with zero glycogen or close to at the end, and then she would have probably done a bit better. But her amino acid concentration is smaller. There is a component, but it's only about 2/3 of the size of the silly males who's run too fast. So glucose is coped pretty well. Glycogen stores are still intact. What that shows you is a micro version of starvation. If you starve, genuinely starve, you would find that you get much more tissue breakdown, you get much more amino acid usage, getting that in there. Right. How are we doing? 5 minutes. Good. We can do diabetic coma in 5 minutes. There are 3 causes of diabetic coma. I tried to get them all onto one slide, and then I decided that neither you nor I would be able to read or understand it, so I've done them in 3 separate slides. Sorry about that. It's cost you an extra page. So there's an osmolarity, osmotic effect, and there's a glucose effect, I think. Yes. Hyperosmolar and hypoglycemic, that's right. Ketoacidotic. You remember I said that you get an increase of production of ketones in type 1 diabetes. If you don't take your insulin when you should, you've got an insulin lack. Usually that's caused by missing a dose. Infections will mess up your whole metabolism anyway, so that you get all sorts of strange things happen. You get fat replaces the glucose. That produces the ketones. That leads to an overall acidosis. Now you've got an acidosis. That produces extreme thirst, which cannot be satisfied. Frequent urination. So again, osmotic and acid effect produces that. Get a high glucose, and you've got your osmotic effect there. Sorry, I'm missing a left bracket, parenthesis. And you get progressive drowsiness and fruity breath. That's the ketone acids, which can be smelled. Smells like slightly rotten apples. Again, it's quite noticeable. If you ever smell it, it's very noticeable. So a good thing to do if you've got somebody who looks as though they're going a bit unconscious, a bit strange, have a little smell of the breath. Good Middle Ages kind of medicine, that is. You could also taste their urine, of course, which is what the Middle Ages medics used to do. You taste the urine, yes, of course they did. If it was sweet, you had diabetes. Didn't know what to do about it, but they knew what it was. You have sweet urine, diabetes mellitus. It means you're producing sweet urine. Right, there you are. So, good old-fashioned medicine. Treatment, you've got to get fluids in, and you've got to get them in fast. So you need to set up a drip, an intravenous line, I think they call it nowadays. You'll see all these television programs. They say drip nurse anymore, no? I always thought that was quite a good one, worth a smack. You will give some insulin to get the blood glucose down, and you will need to give sodium and potassium because of the acidosis. So you really have diabetic coma is dangerous. It can proceed to death quite rapidly. So nasty stuff. So you need to get it. You need serious support. Hyperosmolar, similar effects, occurs much more in type 2s. You get insulin lack. Oh, don't have a dose with. Well, you might. No, you won't. Increased sugar intake. So you overload the system in there, or you've gone the other way and you've decreased fluid intake. Okay, that may deal with it. You can decrease fluid intake and increase dehydration by excess alcohol, of course, which is a diuretic, and increased fluid loss. So high levels of drinking, relatively, not even very high levels of drinking in diabetics is dangerous because of increased fluid loss. Oops, sorry, go back. So you get hyperglycemia in here, hyperosmosis leading to dehydration, collapse of blood pressure, but it has a slow onset, that kind. So you get slow onset over days. So you can generally do something about it. Most diabetics would be able to pick that up with a little bit of experience. They would notice it. Again, to get to the collapsing stage, you need your fluids, insulin, and sodium and potassium, just the same. The hypoglycemic coma, this was the one I was really interested in because it seemed very odd that you should have a hypoglycemic coma, but you do. Either type can get it. You can get by giving yourself an excess insulin dose, and that was used, by the way, as a form of shock. You've heard of electroconvulsive therapy. Insulin shock has been used to treat intractable depressives and things. I'm not sure whether I wouldn't think it's used now. It strikes me as a highly irresponsible thing to do. You give a high dose of insulin, whack the blood sugar down, sends your patient into some kind of conniption, and that's supposed to be terribly good for their brain. Doesn't sound good to me. So if you get excess insulin, if you're a type 1, so you forget, you take an extra dose. Strenuous exercise while not taking any food to replace the glucose. So you're using up the fuel, you're not having a replacement, you can dip too low. Not eating enough. So again, your little old ladies who don't look after themselves, living on a piece of bread and a tomato for lunch, and possibly a little gin. Take some everything is going the wrong way. A little glass of sherry. We never did anybody any harm, a little glass of sherry. Yeah. What is it about little old ladies and sherries? Right. So you go hypoglycemic. That's below about 60. I put in the other units in here because they had the different units in. These are equivalents, exact equivalents. So around about 60, which we set out with our cutoff value for hypoglycemia, you get sweating, you get intense hunger, and confusion. I fall into the last two categories most of the time, personally. But this is a medical emergency, It's a medical emergency. If somebody's in a hypoglycemic coma, you've got to get them to hospital. You've got to get all of those things. You've got to get glucagon into them to start boosting their sugar, and you've got to get glucose into them pronto. And if they're unconscious, you can't give fluids, you'll lose them. I think that is the end. It is. Now, let me say two things to you.